Very much for inviting me back. It's always an honor to come to Michigan. It's my favorite university. It's just that I don't have a job here anymore. I grew up in Ann Arbor, and I will come back, but only virtually. So one of our jobs at NCSE is to promote sustainable campuses. There's an organization for the advancement of sustainable campuses. Some of you may know about it. And one of the things we're calling for is much more virtual seminar giving. I calculated my carbon footprint to come to give you this talk. It was 4,500 pounds of carbon, which I personally put into the atmosphere to travel here. And it seems to me, I do a lot of traveling, and I'm reaching the point where I'm turning down seminar invitations around the world because of my carbon footprint. And I don't know how many of you are ready to do that. Actually, I'm a little tired of traveling, the honest truth. But that said, why not go virtual, for a lot of our talks? It would save Michigan and all the other universities that invite me a lot of money. It would also be good for the environment. And I could spend more time giving these talks if I sit in my office with a camera and a screen, and students could come and talk to me in their offices or in a common conference room. Please investigate the idea of setting up a virtual seminar program. We're going to do that at UCLA, and I think time has come. So that's why I won't come back in person, even though I plan to live to 100. My 73rd birthday is next Tuesday and scares me to death, but I'm still ambulatory, and my brain still works, and I hope I can convince you of that this afternoon. So you have to live a long time if you want to study forests, because trees live a lot longer than we do, and this makes a problem. But one of the nice things about BCI, Barrow, Colorado Island, is that it's on a nutrient-rich soil, and so the dynamics of this forest is awesome. It's turning over incredibly fast, which gives us a lot of handle on the mechanisms of what's going on to control diversity in this forest. So I want to talk to you about that. We started this plot, Robin Foster and I did, in 1980 after I lost a plot in Costa Rica due to the fact that it was on private land, and the owner decided to clear the forest to grow more cattle. I don't eat cattle anymore, so I was a little annoyed. But here's the question that burns a hole in my brain and has for as long as I can remember, and it is this. To what extent are the dynamics of ecological communities dominated by non-stabilizing stochastic forces such as drift versus stabilizing deterministic forces such as density and frequency dependence arising from adaptive species differences, that is, niche differentiation and species interactions? Now, I'd have to say right away that your system may have a different answer than the one I've been studying, and my own colleagues at the Smithsonian and elsewhere don't necessarily fully agree with my interpretation of what I'm going to show you, but that's okay. I'm talking and they're not. Some years ago, Adler and others published a kind of an interesting summary paper of the ideas that I just mentioned verbally. And that was that you could imagine a plane in which you plot niche differences measured by frequency dependence, the strength of frequency dependence on one axis, against the absolute intrinsic rate of increase of the species under zero-sum competitive interactions on the y-axis. And they divided the plane into two sort of major regions, one where competitive exclusion would dominate and the other where coexistence would occur. And they attached to this little graphs of frequency dependence to show you what they meant. So down here is neutral communities which have no frequency dependence, and equal population growth rates. But as you move along the x-axis and also up the y-axis, here are 3 sample points that you tip the frequency dependence, so low frequency and high frequency, the per capita performance of the species growth rate-wise tends to become worse when they get common. These are the two different species, in this case a dotted line and a solid line, getting more and more tipped as you go out the x-axis, but as you go up the y-axis, also a greater separation in the absolute growth rates of the species. So the question that I pose to you is, where do most communities lie, and in particularly, where does the BCI forest community lie on this graph? And I've argued in my book and other places that it's down pretty close to this point right here, pretty neutral. Today I'm going to give some nuance to that, as you'll see. It's a little bit off that point. So for those of you geographically challenged, Panama is a country that is east-west, predominantly east-west. And it's rather confusing when you're on BCI that the sun comes up in the north, which it doesn't, of course, but it feels that way. The canal is right here, and this big lake is artificial, created to provide the water to run the locks of the canal back in the early teens of 1914 rather. And it isolated this piece of hilltop in the middle of this lake called Barrio Colorado Island. And Louis Agassiz sailed his yacht through the canal in the year after it was built and called upon the director of the canal to make this into a reserve. And it became a reserve after that. It was run by Harvard for many years, and then it was taken over by the Smithsonian. So on this island, there is old-growth forest covering the back half of the island. This part of the island, the front part of the island, which faces the canal, which runs here, has the laboratory clearing and was old fields that are now about 100 years old. Sorry, the forest that has recovered is about 100 years old. The 50-hectare plot is shown here, measuring 1 kilometer by half a kilometer in the old-growth forest, except for a lobe of secondary forest right there. And in this, we've censused all plants, as you probably know, woody plants over a stem diameter of 1 centimeter in diameter, which is about the size of your index finger. And we've been following them since the first census in the early years of 1980 to 82 until the present time. Now, I have to say that in its infinite wisdom, for the first time ever, NSF decided not to fund the recensus of the plot, but I thought ahead, and I had talked to the secretary of the Smithsonian that we needed to gazette CTFS plots and have some bridge money in case this happened, which it did. So we are able to complete the census, the 8th census this year, but not on NSF money. And this really irritates me. I wrote to them and I said, how could you fund this for 30 years and not fund it anymore? And they said, well, we didn't like your proposal. Okay. So they didn't like the proposal. But this is a phenomenal data set. You're all welcome to use it. It's publicly available on the web. This is something I'm committed to, is open data sourcing. And as a result of that, there are over 700 publications that make use of the BCI data set. And I'm not just a passive participant here. I don't just census the plot with our assistants, but I actually do analyses. And so some of those you're going to see today. And here's one of the questions that I've been troubled with for a long time. Why are so many tropical tree species so rare? This is a fundamentally unsolved problem. For the first time in my life, I think I might have an explanation for this question, but let me just show you what we're talking about here. It's really something. This was known by Alfred Russel Wallace, but we've had quantitative data only in the last 20 years or so. Here, for example, is a curve showing this phenomenon for the BCI forest. Here are the species on the x-axis ranked from most abundant to least abundant from left to right. And here is their cumulative abundance measured as percentage of individuals. It's sort of interesting that if you consider only the most abundant species, the 9 most abundant species make up half the stems in the forest. So it's kind of like a temperate forest. But look at the bottom half. 153 species make up 0.6% of the individuals. If you go global, we have 20% of all the described tree species in the world in CTFS plots now, 8,500 tree species. half of those species, 4,250, make up 2% of the individuals in the global plot. In fact, it's more amazing than that. There are 600 tree species in the CTFS network that occur as a single individual in the global network of plots. So how the heck are those species making it? It's really, really amazing. So this is the network, and I've just already anticipated my next 3 slides. So we have 8,500 tree species, 20% of the world's trees. 4% of these species make up half the individuals in these plots. And the remainder half, the rarest half, make up just 2%. So why are there so many rare species? But let's go back to the local scale now, to BCI, and ask this question more locally. What controls the composition and commonness and rarity of trees in the BCI tree community? Maybe that'll give us a clue. So if you're already falling asleep, here are my conclusions ahead of time so that I can remind you as I go through the talk. First of all, the BCI tree community shows evidence of both demographic stochasticity and deterministic environmental variance. Second, 2 famous classical hypotheses for maintaining species density and frequency dependence have serious problems as the primary explanations of BCI tree species diversity. Third, and more interestingly, the spatial population dynamics of BCI tree species are very complex, large scale, and inhomogeneous, despite the fact that there's relative homogeneity in the physical environment. And finally, I'm going to propose a hypothesis that is getting a little bit old at this point, the enemy susceptibility hypothesis. I haven't yet published it, but we have a lot of data supporting it, which I will give you at the end of the talk. And this may explain, or I hope it will explain, why so many of these tree species are rare. So to the first point, evidence of demographic stochasticity and deterministic environmental variance. Just to define terms here, I use the term demographic variance or demographic stochasticity to describe changes in population abundance due to chance differences among individuals in birth and death rates. So that's familiar to you from population genetics, the same idea. Rare species ought to be more subject to demographic stochasticity. In contrast, I use the term environmental variance to describe changes in population abundance caused by all other non-stationary physical and biotic processes that cause correlations among individuals in birth and death rates. So here we don't have correlations, here we have correlations. That's a fundamental distinction. So because of the fact that rare populations may have an imbalance between birth and death rates among the individuals, you'll find that populations change in abundance due to demographic stochasticity, and less so in common species. So under that idea, we'd expect these very rare species that make up half the community to be much more subject to demographic stochasticity than the common species that are extremely abundant. So we modeled the intrinsic rate of increase in the classic way. The little r is equal to a j. I shouldn't have used j, it's time, but I didn't think about that. Anyway, it's the log of the ratio of the change in population abundance divided by the time interval. And we did not assume carrying capacities, and you'll see why not in a minute, but we modeled in a discrete fashion the change in numbers of a species with positive terms for birth as a Poisson process and a negative term for death rate as a binomial process. And this gets plugged into a model where we can calculate all of these demographic parameters. And since we expect demographic stochasticity to be smaller, or larger rather, in populations of small numbers of individuals where we would expect no correlations between birth and death intervals, we'll see if that in fact is true. Engen and Landy some years ago wrote a book on stochastic population dynamics and conservation, in which they pointed out that it's possible to differentiate the changes in population size that are due to demographic variance or demographic stochasticity from those that are due to environmental variance. And the really neat thing is that there's a power law of the relationship between variance in little r, or absolute value of little r, that is a power law with abundance. So, and it should have a slope of minus 0.5. And whereas environmental variance should have no slope with the absolute value of little r. And so we calculated that over the 6 census intervals that we had at the time. And what you can see here, here's a pure environmental stochasticity against the slope of absolute value of r on species abundance. And down here is the minus slope of 0.5 for pure demographic stochasticity. And so we have a mixture situation here. We have generally about minus 0.3 to minus 0.4. So it's closer to the demographic stochasticity line than it is to the environmental stochasticity line. But there's some jumping around here. And one of the things that's characteristic of these is that the more deterministic cases here and here were both El Nino droughts that caused massive mortality in the canopy layer in the forest. But it didn't shift it too much. So it's got a heavy component of demographic stochasticity as well as a component of deterministic stochasticity. If you look at from the modeling that we did for the neutral theory, taking the values of observed birth and death rates, the neutral theory predicts this peak right here around 0 intrinsic rate of increase, the red line. But the observed distribution of little r values for the BCI tree species peaks at the same point, but it shows some deviation from the wings of the tails of the distribution. A little bit more excess declines and excess increases. But it doesn't do too badly here. It captures a good chunk of the distribution. All right, what about evidence for the mechanisms for stabilization in this forest? And there are 2 mechanisms I'd like to describe. The first one is the classic Janzen-Connell mechanism, which all of you have probably learned. And the basic idea is very simple. This was proposed by Dan Janzen in 1970, and Joe Connell wrote a similar paper a year later. And what I want to pose is this question: does this mechanism have the potential to regulate the number of reproductive adults in the populations of BCI tree species? Now, originally when this mechanism was proposed, the primary focus of interest was in maintenance of diversity rather than regulation of the abundance of populations, but the two have to go hand in hand. So here's basically the idea, if you don't remember it. Basically, we have a mother tree here that drops most of the seeds below itself, and this is the seed dispersal kernel. So most of them fall under the parent, and only a few get transported a long way away. However, because there's intense seed and seedling predation, the ones that are under the mother tree don't survive, and so this is the probability of survival of an individual seedling. And so you multiply these 2 curves together, the recruitment of new saplings will be at some displacement distance away from the mother. And so the idea was that this would open up the places where mature individuals of species A were to the colonization by any other species. But the idea was that it had to be dependent on host-specific enemies. Turns out that's not true. Because of the aggregation of the seeds, if you had a randomly mowing robotic lawnmower going through the forest, it would cause Janzen-Connell type effects. So it doesn't depend on host specificity whatsoever. The problems are not that with the model are not that, however. So in order to study density dependence, in the late 80s, Joe Wright pointed out to me that we were missing much of the dynamics because we didn't have any data on seed rain and seedling germination. So we set up a grid of 200, not a grid, but a network of plots that were half square meter in area. And below this trap or nearby, there were 3 1 square meter seedling plots. So we could, if we make the assumption that the seed rain into the seedling plots was the same as in the trap on a per unit area, we could examine the relationship between the number of seeds falling in here and the number germinating here to get a measure of density dependence. And my student Kyle Harms analyzed these data, published a very prominent paper in Nature on this. And basically the idea was, if you plot the log of, call this the seed rain at stage I, and then the next stage seedling output on this axis, again log transformed, you'd take log in order to get rid of the effect of the mean, because this axis is on a very different scale than this one. You can find, if you find slopes of 1, there's no per capita density dependence, but if the slopes are much less than 1, it indicates that the more seeds you put in, the fewer per capita seeds that you get out. And these are the results across the seed traps. So here's the seed density in numbers per meter squared per year. And you can see it's a very different scale on the two axes. And this axis, this line has a slope very much less than 1. So very strong density dependence. You say, aha, the smoking gun, we proved that this is the mechanism regulating tree diversity. Hold on a minute. But here's another graph from his paper showing that none of the slopes for the individual species curves even nearly approach 1, where 1 is out here somewhere. Most of the slopes are down around 0 or even negative, meaning really strong effects. Well, the problem with this hypothesis, before I say that, there's one more thing I want to tell you. My student Yifeng Sun analyzed the recruitment data over the past 30 years for tree species in the canopy layer, and he found that basically 75% of them had essentially no regeneration, no replacement of themselves within 5 meters of an adult. And even those species that did replace themselves, it was usually a very low rate down here. So everything looks good, right? Well, here's the beginning of the problem. This is a graph showing the effect of adding one conspecific neighbor to an annular distance around a focal plant on the growth rate of that focal plant over a 10-year period, the relative growth rate over a 10-year period. So on the x-axis, I have the diameter of the focal plant. So if you're small and the plant you insert in this neighborhood is nearby, less than 5 meters away, it has a huge depressing effect on growth rate. But look what happens as you go to greater and greater distances. By the time you're out to 20 meters, we're very close to the line of no effect. And it goes essentially, if I plotted 20 to 30 meters, it's essentially no effect at all. Now, big plants are relatively indifferent to the addition of a conspecific, which in this case was a randomly chosen size class. So it's not surprising they all converge on the line of no effect. But the point I'm making here is that these Janzen-Connell effects are essentially very local. They're defined within, you can detect them within about 20 meters of a given point, but not beyond. And the same thing is true for survival. I just didn't show the graph. So the question then becomes, conventional wisdom says that this is sufficient to regulate the adult population. And that was the conclusion from the Harms et al. Paper. But I think there's reason to doubt that. So very strong and pervasive Janzen-Connell effects on a local basis, but very, very local. Okay. Now here's the catch. I want you to imagine that none of the seed is dispersed. It falls only under the mother tree. Now suppose mother trees produce 1 million seeds and only one survives to replace the mother. Then you have very strong density dependence under the mother. Or it produces 1 seed over its lifetime and it replaces the mother. Very weak density dependence. It doesn't matter what the strength of density dependence is if none of those seeds disperse away from the crown of the mother tree. So density dependence that is restricted to the progeny of single trees beneath the mother's crown cannot regulate the number of adults in the population. It's impossible. So non-regulatory within progeny array density dependence accounts for almost all of the reported density dependence. In Kyle Harms' study, I went back. He hadn't looked at where the adults were of the trees that were being trapped in the seed traps. And it turns out if you take out the ones that were landing under mother, you lose 90% of the density dependence that he found. So that's scenario one. No seed dispersal, species all beneath the maternal parent, replacement of the parent by individual of the same species next generation, regardless of the number of seeds produced in the parent tree. So you could have 1,000 parent trees or 2 parent trees, and if they're all doing this, then they just neutrally replace each other, and there's no regulation of the adult population. So that's one scenario with no dispersal. Let's go to the opposite extreme with infinite seed dispersal. So let's say that there are S species equally abundant, just for sake of argument, and that any species can regenerate under anybody else but itself. So it's pure Janzen-Connell. So I cannot regenerate under I, but it can regenerate under any J. So what sort of limitation does that impose on the growth rate of the species in question? Not very much, because it's got S minus 1 over S sites that it can regenerate in, and only 1 site that it can't regenerate in. So, or 1 over S. So essentially, this is a very weak dynamic constraint on the regulation of adult numbers. The other mechanism that I want to talk about is equally famous. It's the Chesson-Warner storage hypothesis of frequency dependence. And if you don't remember this idea, there was a paper published in American Naturalist in 1981 by Bob Warner and Peter Chesson, proposing a lottery mechanism for the maintenance of species richness through rare species advantage. And I didn't initially understand the paper until Bob explained it to me with a cartoon, and this is the cartoon. He worked on pomacentrid reef fish in Australia, and he said, imagine 2 of these fish which have interspecific territoriality and they feed on corals. They essentially have the same ecology. One of them's common. There are 100 territories here, with 90 belonging to the blue species and 10 belonging to the yellow species. And let's imagine that the mortality is the same in both species, so let's say 10%. So after a mortality event, there are 81 left of the blue species and 9 left of the yellow species. But now let's make there be asynchronous reproduction in the 2 species, so that the rare one is the only one reproducing the next year. Well, then it gets all the sites that were vacated by the blue species, so it grows to 19 individuals, but the common one has only fallen to 81 still. So there's very strong rebound in the rare species in this case, and this kind of frequency dependence will work to maintain diversity, and it's very strong if you have a big difference in the recruitment times for the species. But there's a problem in applying this to BCI tree species, which I will show you in a minute. But we thought, oh boy, Liza Kamita and I have been looking at seedling communities. We have 20,000 seedling plots in the BCI forest. Every 5 meters there's a 1 square meter plot, so we have a 4% sample. And so we have 3 of these plots under virtually every tree in the forest. And so we have great detail on the germination and subsequent seedling survival of these species. Here's an example pair of years with a difference score in terms of the percent change from 1 year to the next. This was a very good year for these species, bad year for these. But what's interesting is there was almost no correlation between in the same year for seedling survival thereafter. So germination cues may be different from what allows the plants to survive or not. But in any event, there's a big problem beyond this irregularity. And here it is. Basically, the assumption of the storage hypothesis is that there's asynchronous or pulses of recruitment which propagate as a traveling wave through age and time to the adult stage. And if these are asynchronous between 2 species, that can allow for coexistence. So this whole wave moves along, and so here's a time 2. But the assumption this is making is that this is also true of size. And that's where the problem lies in this model. Because if you look at cohorts of even-aged saplings, they lose coherence as a group of similar sized plants over time. So here is our most common canopy tree species. This is the distribution of diameters of plants that were born in, or that entered the census in the same year in 1985, and were all between 10 and 20 centimeters. So they were all down here. And so this is by 2010 rather, they had distributed their sizes spread out over this variation. And here's another species. They all look like this. They're basically totally spread out. So these are all equal aged saplings. And this is an amazing species. It had some that grew by 800%. Now, if you stack these up and ask how do different age cohorts look, here, for example, is Pharamia. And the different colored lines here are different age cohorts. So the blue line are 25-year-old plants. The green line are 20-year-old plants. The 15-year-old plants are the red line, or 10-year-old rather, and the yellow are 15-year-old. They're essentially pretty much superimposed on one another. So basically, I'm trying to do this mathematically. It's a complex problem in partial differential equations, and I'm having to learn my PDEs again. I'm a little bit behind the times, but in any event, I think this is going to be a real serious problem for the Chesson model, because there's a total loss of coherence of the seedlings as they move through age. So what I conclude is that frequency dependence, or rare species advantage, is required. Oh, this isn't a conclusion. This is just true. Frequency dependence is required by all equilibrium theories for the maintenance of species diversity. And a leading hypothesis for frequency dependence is the Chesson-Werner storage hypothesis. However, a major assumption of this hypothesis is that age cohorts maintain their coherence until the adult stages are reached. And that's a long time for these trees. We're talking about 100 years or longer, 120 years. And I think what I've shown you is that this assumption is seriously challenged by the BCI data. But we can cut to the chase here and ask, what is the empirical evidence for frequency dependence in the BCI tree community at the whole plot scale? I'm interested in the maintenance of diversity within the 50 hectares. And the basic idea was captured in this graph that was published by Volkov a long time ago. But basically, what you expect is that the ratio of the per capita birth rate to the death rate ought to exceed 1 for rare species and fall below 1 for common species. And in the theory we developed, this was a measure of density dependence, this C parameter, which determines the shape of this frequency dependence curve. But anyway, that qualitative shape should be the case. You should find greater than 1 birth rate to death rates for rare species, and for common ones you ought to have the opposite. And here are the data for, except for the last census. Basically, over 4 orders of magnitude in change in starting abundance, from over 10,000 to 1 to 3. This is 1 standard deviation, so none of these lines deviate significantly from a replacement. Not good evidence for frequency dependence. So we recently published a paper 2 years ago showing that immigration rate is sufficient in the plot of rare species to maintain diversity without invoking density and frequency dependence. That was published in PLOS One, and Rick Condit was the lead author on that paper. Now, you could have an alternative explanation, and that is the lack of frequency dependence in per capita birth and death rates could be because BCI tree species are already at or near their carrying capacities. And remember, we didn't include carrying capacity before, and I'm going to show you why in a second. However, as we will now see, the dynamics of BCI tree species populations are inconsistent with this alternative explanation. And that's because the forest is really far from any equilibrium. Just an example to tell you that in the last 30 years, 1/3 of the species in the BCI plot have changed by more than 50% in total abundance. 1/3, and this includes common species, rare species, shade-tolerant species, shade-intolerant species. Everybody shows incredibly fast dynamics. So that leads me to the third part of my talk, which is that the spatial population dynamics of BCI tree species are complex, large-scale, and inhomogeneous, despite the fact that we tried our best to get the most homogeneous physical environment that we could on the island. We failed because there were hills, and we are glad we have some hills, but the bottom line is we have a very complex story to explain. So what I did in order to display what I'm going to show you is that I divided each population of a species into 200 quarter-hectare plots and measured the intrinsic rate of increase of the species over one census interval. And then I made a map of the changes in the intrinsic rate of increase over time to make a movie for you. And what these show is that fine-scale Janzen-Connell dynamics are simply cannot explain what we see, because they would explain a very fine-scale pattern of homogeneous dynamics, because it's very local, remember. But that's not what we see. In fact, what we see is very, very large-scale patterns that we're just capturing the edge of in 50 hectares. So I'm going to tell you in a moment that 50 hectares is not big enough. We have to go much bigger. So here's one of our tree species, and let me tell you how to read this graph. Each color, the red colors indicate intrinsic rates of increase that are above at the maximum level in the range of intrinsic rates for the species, and they're positive. The light greens and yellows are right around zero change, and down here the blues and violets are strong negative decreases. So this is rate of change you're looking at. So the movie I'm going to show you is essentially the second derivative of population change. It's a measure of the sensitivity. It's the acceleration, if you like, of the population. So watch the patterns, recognizing that this is about 160 feet on a side, each one of these squares, so they're not trivial. They're definitely much larger than the spatial scale of 20 meters around an individual tree. So now I'm going to run the movie. And in order to make a movie, there are 7 censuses, so I had to interpolate the colors, which I did in R, but you'll just see that there's very large landscape scale patterns going on. Many of these populations show an initial flush of red because we had that El Nino event early in the history of the plot, and so there was a lot of recruitment in the understory, and so a lot of the reds were due to the increase in light that was in the understory. But as time went on, the populations changed their patterns. So the patches of red and blue changed dramatically in these plots. This is Hybanthus, our most common understory treelet, and this shows the huge amount of red, a lot of positive growth early in the history, but again due to the opening of the canopy. But then it had a massive die-off across the plot, which we cannot yet explain. So this graph turns largely blue halfway through it, as you'll see. And what you'll see is that the spatial scale is huge. So it hasn't yet started to turn blue, but now it's going to turn really blue. So if you're colorblind, at least you can see the blue. And now it's starting to recover. You see a few more patches of yellows and reds coming back, but it's fallen from about 40,000 individuals to a little more than 20,000 in the course of these censuses. This is a gap species, Cecropia insignis, and it has a very sort of Syrah pointillist pattern because it's dependent on light gaps, and so that shows up fairly well in this movie. So you can see some of the life history characteristics of species in these spatial dynamics. And these match pretty well where we have tree fall gaps. So big disturbance here, as you might imagine. These aren't Janzen-Connell. This is something else. And here's a shade-tolerant species in the Lauraceae with great big seeds that are dispersed by toucans. Again, a huge flush down here, which goes away, as you'll see. So early in the sequence, there was big recruitment there. And then it just goes away. So what's explaining these patterns? Aha. What is indeed explaining these patterns? Well, just let me recap this briefly by pointing out that if you take snapshots of the intrinsic rate of increase for species, they all look like this. They look like exploding little patches of populations that have little red. This is a high growth rate area. 10 years later, there's no matchup between where there was rapid growth here and over here. Same way with this species, same way with this species. And the patches of decline are also moving around. So these are incredibly spatially dynamic populations. And I've done a lot of analyses of these, but I'm not going to go through all this. I talked about some of this 5 years ago, and I haven't done a whole lot more with it since then, except for make the movies and start to analyze the spatial patterning. But this is the boiling mud pot model of population dynamics. So if you go to Yellowstone, you see things like this. And this is precisely what it looks like. You have sort of explosive growth here and then explosive growth over here. And this doesn't look like regulated populations, folks. I mean, this looks pretty unregulated. So what is driving these population dynamics? And can it explain rarity in the forest? Well, I have stabbed at this with my enemy susceptibility hypothesis, so let me give you a heads up on what we think might be going on. We haven't proven it by any means, but here's the idea. So we've discovered that a major cause of tree death is caused by heart rot and root rot fungi that hollow out the tree trunks or weaken the roots, and so you get tip-ups or snap-offs. And so the boiling mud pot dynamics, I'm hypothesizing, are due to spatially and temporal lagged predator-prey dynamics between the tree host and their fungal pathogens. And the trees disperse and local stands increase in abundance before fungal pathogens discover them, infect them, and then it takes decades for the infected trees to die as their trees are being hollowed out, and they eventually get too weak to stand up and they get blown over in a windstorm. Now, what I'm imagining is that most tropical tree species are rare because they are more susceptible to heart rot fungi, or maybe they also share more of these fungi with other tree species than common tree species do. Therefore, rare species have fewer safe sites in which to regenerate than common species do, because there are fewer places in the forest that they can grow that are far from the source of fungal contagion, because they also can be infected by heterospecific from trees that are heterospecific as well as conspecifics, whereas the conspecifics only get infected if they're next to an infected conspecific. So leave it to the Germans. They invented portable CAT scan equipment for taking tomography data on trees. And so you take your portable laptop out into the forest and you strap on these sensors, and then your laptop displays a cross-sectional area showing you where there is heart rot in the tree. So we've done this on about 2,000 individuals now. all the rare species and the really rare ones, and a subsample of the common species. And the hypothesis would be that rare tree species would have heart rot at a smaller diameter and a younger age than the common species were. And that's precisely what we found. This is the statistics on it. So basically, it's the opposite of the rare species advantage notion. If you imagine that heart rot increased with abundance, then you would have a graph like this. But this is, in fact, what we see, that heart rot is greater in the rare species than in the common species. And here are the kinds of data that we have to show that. These are logistic regressions. And this is tree species abundance above 20 centimeters. This is the probability of heart rot. Here are trees that are 20 centimeters, 40, and really large, over 60. There are very few rare species that make it to these large diameters. So what this tells us is that if you get really big and you're really old, you're almost always going to have heart rot. There's nobody going to escape heart rot. But the ones that the rare species never make it beyond about 20 to 30 centimeters, and so they are more infected by these heart rot fungi. We've also we have a grant to identify the fungal species molecularly. So when we find a hollow tree, we core it and take out the fungi, culture them up, and then we test for we molecularly identify them, and then test for their host ranges on sawdust cultures. And what we're finding is, number one, that more species are found in rare trees than in common, more fungal pathogens, that is. And the fungal pathogens found in rare species, the few species we've tested have broader ranges than the ones we've tested from common species. So far, so good. But it's only in the middle of the grant, and we have a lot more to do. One of the things we're doing is testing the Koch's postulates. So we infect a toothpick and we insert it in a greenhouse plant. We have a large array of saplings from the trees on BCI, and we take when it gets big enough to have heartwood, we slit open the bark and insert the infected toothpick into the heartwood and see if the fungus spreads out into the tree. And so we can test for host ranges in this way, and we haven't done that yet. The final thing we're doing is analyzing the enzymes that break down lignin in the fungi. The fungi are in an evolutionary arms race with the tree hosts. The hosts produce lignin, and it's very diverse. And the fungi can either break it down or not. There needs to be a one-to-one matching of enzymes with lignin varieties. And so the hypothesis is that the rare species have broader host range fungi that have a broader range also of lignases to attack the variety of hosts that they do infect. So I'm wondering, could this in fact be a general explanation for rarity across the tropics? So if we succeed in showing that it seems to be true for BCI, we plan on doing it in very different forests phylogenetically to see if it's likely true there as well. And I think I've talked long enough. So there are my take-home messages, and I'll take any questions if there's time. There isn't. Yeah.
I thought it would be interesting to plot the distribution of your rare and common species on angiosperm phylogeny to see how, if we have any pattern.
No, the abundance is not phylogenetically conserved. So rare species are well distributed across the angiosperm phylogeny. Rarity is not really a trait. It's sort of the result of your good or bad luck and your site requirements and all kinds of other things. And this doesn't seem to have any phylogenetic signature at all. That is not to say that the fungi that infect them are not phylogenetically, that is, there's a matchup of clades of fungal species with their hosts. So there's some coevolution there.
But still, say if you look at the 9th species that occupy, say, half of the individuals of your community, to see whether they are basal or derived on angiosperm phylogeny, but to see if they are associated with clades, whether basal or derived.
They're not.
Really diversified.
No, the answer, I can answer that definitively, no. Not true. But I mean, we were very interested in that question. But commonness and rarity has very little phylogenetic signature. I'm not saying it has none, but certainly within the BCI plot, it doesn't have any. Yeah.
They keep a consistent effect of fungal susceptibility on some of these species. Wouldn't you expect, I guess, that extrapolating deeper, if you're actually measuring an effect over ecological timescales, I mean, doesn't that have some predictions for extinction of these things, and why aren't we? I mean, it seems like you would actually, extrapolating over deeper timescales, you would actually lose a heck of a lot of this diversity if there's that.
The answer is you've hit on a really important point, and that is that frequency dependence is the classic mechanism to maintain rare species in the system. The fact that we don't detect it, and we only detect maintenance of diversity by immigration, suggests two things. One is that we're not looking at the right spatial scale to find where these species are truly abundant and adapted. We have no idea if these rare species in our plots are rare everywhere. That's a huge question that is unanswered. So the possibility exists that they're doing just fine, thank you, somewhere else. However, theoretical models going back to MacArthur show that fugitive species can be maintained in the system just by dispersal. And with dispersal limitation, you have lots of opportunities for inferior competitors to stay in the very long, evolutionarily long, within these systems without going extinct. So coexistence turns out to be a very slippery thing to define. And I talk about cohabitation. I don't talk about coexistence because I don't know what coexistence is. Yeah.
So following up on that question, could these rare species over evolutionary time be evolving resistance to pathogens, to their fungal pathogens, and then the common species eventually get a pathogen that they can't overcome, and so we kind of see that the common species.
I think that's very likely. I think this, I was supposed to repeat the questions. Is everybody able to hear the question? The question was, over evolutionary time, will species become adapted to their fungal pathogens and become resistant to them? And the answer is that anytime they evolve a new side group on a lignin molecule, they become resistant to the specialized enzymes that break down the precursor. So yes, this is probably, there's evidence of evolutionary arms race. If you look at the rapidity of genome evolution in angiosperms and in fungal pathogens, the housekeeping genes are evolving at one rate, but the fungal fighting pathogens are evolving 10,000 times faster. So there's very strong evidence for coevolutionary arms races here.
How do you distinguish between, I guess, the possibility that really you're looking at some of these rare species are actually essentially ecologically marginalized within, unmeasured aspects of the 50 hectare study plot. And so you're really picking up the outcome of species that are sort of mismatched to that landscape on some particular spatial scale.
Well, we've looked for niche differentiation in a variety of ways for habitat limitation. So for example, pioneer species that are gap dependent will always be rare in the system because the gaps only constitute about 2 to 5% of the forest, depending on the census year. So that's one example of a specialization. It's not limited to a particular topography or hydrology, but the vast majority of rare species show no signature that relates them to any physical environmental variable that we've been able to measure. Now that's not to say that those don't exist, but at the spatial scale of 50 hectares, we can't find it. So that doesn't answer your question. It just puts it off for the future. But, the classical hypothesis for rarity is specialization. And it's very hard to find a signal of that. I've answered all your questions. That's wonderful. Yeah.
Rarity is common microbial world too, and a lot of microbes have either specialized or generalized viruses. Well, I mean, would this same might this apply to the microbial world as well? The enemy susceptibility?
Well, I sure hope.
Yeah.
No, I have no idea. You tell me.
I like it. no data, though.
No, I mean, we're just at the very beginning of this. I mean, the idea is only about 4 years old now, and we're in the middle of a Dimensions of Biodiversity grant, and we don't have any smoking guns saying you're totally wrong yet, but I think it's going to be a nuanced answer. It's not going to be as simplistic as the way I've formulated it for you today. It'll be more nuanced, but at least at this point, the gross predictions are still holding up.
But do you see it in temperate forests that have high diversity, like in the Appalachians?
Why don't you go tell me? Find out.
I mean, do people know anything about the root rot and whether the more rare species there are more susceptible to?
As far as I know, we're the first person to ever propose this crazy idea. And so, no, it's not been tested anywhere else. So I'd love to see that be done.
So how far are you into writing a model or running simulations that show that method could happen?
Well, as you ask, Jeff, where are you? Okay, there in the back. We're collaborating on doing a simulation model. It's not very far along, but we'll let you know in a month or two what we find out. But these simulations are very slow. I have a one species, one pathogen model, which shows limit cycle behavior under certain parameter conditions. So I get populations fluxing and migrating out, and then the pathogen disappears, and then recolonization. And so you can, it's done on a sphere. I guess I did it on a two-dimensional strap, so no edge effects. And I can, under the appropriate space and time constants for the dispersal of the fungus and the tree, I can get situations where the dynamics are balanced and you get a fluctuation, predator-prey cycle, spatially. But I don't know, I haven't done any modeling yet of the coupled population dynamics of species that share pathogens yet. So I've just got initial stages of the simulations done. And I don't think it's going to be analytically tractable, but I may be wrong, maybe. Yeah.
Problems of the enemy susceptibility hypothesis if we consider that in the evolutionary arms race between the parasite and the host, that the rare species are at a constant double disadvantage because they're so rare and their numbers of propagules are small, so they have the effect of their ability to keep up in that race must be hindered by that.
Agreed. This is the opposite of rare species advantage. It's a rare species disadvantage. And so what we've shown in the BCI plot is that these species do, in fact, go locally extinct frequently, and they re-emigrate from somewhere we don't know where.
So the hypothesis explains the balance of commonness and rarity as a transient effect, not as a...
Well, except that if you have immigration in a fugitive species sense, you can show with dispersal limitation, you can still maintain those species, but they never get abundant. So with dispersal limitation, they can stay in the system, but there has to be aphasic relationships as population. You can't have everybody being depressed everywhere at the same time. So you're right, if they're all being driven down everywhere at the same rate, then those species go extinct globally. But if they're out of phase at different parts of the range, and we show, I didn't present those graphs, but let me see if I can get to them. Okay. This shows the Moran effect in, let's see, what did I do with the... What you can look at is the spatial autocorrelation and the intrinsic rate of increase in these populations. And very common species show essentially positive correlation in fluctuations of populations near each other. And that's called the Moran effect. But as you get further away out to 100 to 200 meters, that disappears more or less completely. But if you look at the rare species, you find sigmoidal curves where they're positively correlated close on. At intermediate distances, they're uncorrelated. And then they become negatively correlated at large distances, suggesting that these are fluctuating in antiphase. Okay, so what I'm imagining is that you have a much larger landscapes. You have populations doing this of the rare species. And But the common ones may be also doing that, but their spatial scaling is like tens to hundreds of kilometers. Okay, so maybe they go negative, across Panama, but not within the BCI plot. So that's all I can say to your question right now. But anyway.
Need to wrap up.
Okay.
Thank you.
Thank you. Lots to talk about.